Welcome to the presentation Plants as a Source for Drug Discovery. First of all, I need to say thanks to the International Pharmaceutical Students Federation for this nice invitation. The contents that I'm going to be discussing with you today are, first of all, a general aspect of strategies for drug discovery from plants, and then I'm going to share my own experiences with epanorine, which is a lichen metabolite, some experiments that I have performed with my students about Buddleja globosa hops through an extract called BG126, bioactives from agro-industrial residues like RAPS polysaccharides, as well as artichokes bioactives. First of all, I took this nice scheme from a publication recently made from Productos, in which they divide this scheme in the upper part, which is the traditional approach for studying plants that have a tradition in ethnopharmacology or ethnobotany. And then researchers take plants and by some sort of trial and error start making extracts, crude extracts, and then they can go ahead and get compounds for experimental screening in first in vitro experimental screening, and also with the isolated or some more refined compounds experimental screening, and in finding some sort of results that can show that ethnopharmacology or ethnobotany have a rationale for this traditional medicinal use of natural products. Later on, with computational resources that give rise to some sort of in silico screening with QSAR and docking experiments, researchers can find compounds that are found in plants. And then with this, we could call it backwards approach, can get some experimental validation for the ethnopharmacological use of certain medicinal plants. So both approaches are still useful. I need to declare that I use the one more traditional from the superior part of this diagram in my research. Also, if we put attention on this graph that's taken from a publication on Journal Natural Products, and that shows approval of drugs for use by regulatory agencies since the 80s till more recent years, 2019. And we analyzed this in using these codes that natural products are in dark green, and in a lighter green natural botanic products, and in lighter green synthesis product, starting by natural inspired products, and in red totally synthetic products. You can appreciate that even in more recent years, the totally synthetic approach is not more important than products that in some way are related with natural products. So that's a very important truth that one have to have in mind if interested in drug discovery. Nature is inspiring. And if we look at the scientific interested in natural products that is inspired in ethnopharmacy or traditional use of medicinal herbs, you can see how starting in the 50s of the past century until a month ago or a little bit more, there is an increasing interest in this area. And also I would like to show a personal experience with a magazine that used to be circulating in the 90s in my country. And this was the past century, and they already were putting issues or articles showing that natural products are important to drug discovery. In addition, the Chilean government, through the Ministry of Health, put a few years ago a decree that declared 103 medicinal species as herb medicines. And there is this booklet that describes the traditional use of all of these herbs. So there is a recognition of these herbs as potentially useful drugs for treating different diseases, as the traditional use shows. So I'm gonna now start showing some of my work, which is a teamwork with the students and colleagues. And we put this paper a few years ago about one lichen metabolite. Our School of Pharmacy has a long story of the study in the area of lichens. Professor Juan de Quilot and his disciple Cecilia Rubio have been working on this for several decades. And then we took one metabolite that is epanorine, that comes from this very yellow lichen that grows attached to the rock, and it's really hard to relieve from there. Um, in this slide, you can see how the cells treated with epanorine look like under the microscope. In A, you see cells growing in cell growth media with no treatment. B represent 28 micromolar of epanorine, and you see you can recognize the loss of cells. Then D is the solvent, dimethyl sulfoxide, which is, even though it causes some damage, is not so much as the epanorine. And then we have tamoxifen that causes some, but not too much visible damage to the cells. And E is ethanol, which is the solvent for tamoxifen. We also wanted to know whether is oxidative stress a mediator of this toxicity or cytotoxicity to the epanorine. So we made an assay of a mitosox, which is a sensor of oxidative stress. We used as positive control doxorubicin, which is known to cause oxidative stress on the cells, and it gave us a signal, a fluorescence signal that was not detected neither by the solvent or by epanorine. So the damage of the cells caused by epanorine was not mediated by oxidative stress. We did a cell cycle study by flow cytometry, and we could detect that cells treated with epanorine had a G0 arrest. If you compare with solvent, you can see here BC that the cells are undergoing S phase and G2M cells, which are diminished when they are treated with epanorine. Tamoxifen also causes this sort of effect, since we could see here the solvent of tamoxifen and cells that are in S and G2M phase, and here cells growing happily in cell growth media had cells a number of events on S and G2M phase. So we could understand that part of the mechanism was cell cycle arrest. Was apoptosis part of the mechanism of epanorine killing MCF7 cells? We performed this assay, and by the tunnel assay, in which the 3' fragments of DNA are stained by a short polymerization at these ends with a uridine that is marked with a fluorochrome. And so if the assay is positive, you could see green stained cells, which we only could detect with high concentrations of tamoxifen. The solvent controls didn't show anything, neither epanorine. So we suppose that this was an apoptosis independent mechanism, and then we need to perform other studies to confirm this observation. Then I'm going to pass on Buddleja globosa hope, which is called Matico. Here you can see an image. This is a small bush with flowers that are like globes. They could be yellow, orange, or even white. In the traditional uses of this medicinal plant are stomach ulcers, stomach aches, traumatism, and wound healing. I'm performing several studies on wound healing that I'm not going to show in this presentation, since there are patents pending, so I cannot talk too much about that. I'm going to show some analysis that we have done more related with their anti-cancer activity of the extract of Buddleja globosa. I need to say that we are working with the special extract of Buddleja globosa, which is an hydroalcoholic extract that is donated by Laboratorios Jimena Polanco. This is a patented extract. And what I'm showing now is part of the undergrad thesis of Jordi Madariaga. He worked with cells that are called cell lines that are called AGS, which are from gastric cancer, and their counterpart that are epithelial cells but normal. And he tested a variety of a long range of concentration of this extract, and look at the viability by an assay that is called resarsurine. And then this graph shows the logarithm of the concentration, and the viability. Very interesting, very low concentration, show even a pro viability activity. Then there is in the middle some inhibition that shows no great differences between both cell lines, and I need to explain that we want to have a large difference between normal epithelial cells that I showed previously with epanorine versus the cancer cells, and only at higher concentration we can differentiate the effect, which the just one cell line, which is gastric epithelial non-cancer cells, get constant or a plateau on the inhibition of viability, whereas cancer cells go with a higher, near 100% inhibition of viability. So these show very interesting results that obviously need to be confirmed. We made some molecular studies thinking on a potential activity on cell cycle arrest. So we looked at two molecules that are very important, which are p53 and p21, which are regulator of the cell cycle. And we looked at the expression at the mRNA level to see if the extract caused any changes. This was performed by a conventional PCR and is pending to do it by quantitative PCR to be able to check on these differences, which are only at this point made on the basis of densitometry assays of the gels. Anyways, P53 didn't look like it was altered or changed, but the extract. However, P53 at certain concentrations showed an increased activity which is consistent with cell cycle arrest. Obviously, as I said, this needs further experiments. I'm going to change gears here and go to another way to obtain results or extracts that can be very promising and very important and are within the area of circular economy, and it's related with the recovery of bioactives from different residues, in this case from the agro industry. So one of the cases I would like to show today is related with wraps. We have a very high production of canola oil, which is obtained from wraps. And the problem here is a byproduct, which is called the rapeseed cake, which is a very low value. Economically talking, it's used to feed animals. And what we did here with a colleague, Dr. Concha, and also an undergrad student, COPS, was to obtain polysaccharides, treating these waste, this residue, with enzymes that allow it to have an extract of pectins. And these pectins were evaluated of their activity on different systems. Here I'm showing the work of Mauricio Reina on his thesis for obtaining a master's in bioactivity of natural and synthesis products from our university. So he took some of these extracts and he did a very interesting work starting with different raw materials from different vendors of these rapeseed cakes. And he used a neuroblastoma cell line called SHSY5Y and found an interesting cytotoxicity, and he calculated the concentration called IC50, which is the concentration needed to kill 50% of the cells in the cell culture. And here are the values, which were statistically identical. There were no significant differences between the vendors. And then he did the experiment on fibroblasts, normal fibroblasts, and calculated also the IC50. And if you can see only the numbers, you need a lot more extract to kill fibroblasts than to kill the neuroblastoma cells. And when you did the calculation to have a selectivity index, it's just a ratio between the IC50s, you find between 3 to 6 times selectivity. Is more selective, this product, this polysaccharides or pectin mixture over the cancer cells as compared with the normal cells. So this makes this product very promising. We did a lot of mechanical study, and this is a beautiful essay in which the hypothesis is that these pectins act by binding to a protein called galectin. Galectin, which is a marker of malignity. The more galectin a cancer expresses, the worse is for the patient. So we made an inhibition experiment in which we put in the culture media a binding protein to galectin, basically blocking the galectin and making the same proliferation assay. So when there was no galectin on the media, these different extracts coming from the different vendors made their job of inhibiting the proliferation. However, when we started to increase the concentration of this blocker, the galectin-3 binding protein, the effect of these extracts was abolished. So here is an important proof of mechanism of action of this extract. In addition, we were also interested in understanding if apoptosis was involved, and we had an essay to measure caspase's activity, and we found that all of the extract coming from different raw materials were able to activate caspase activity, giving evidence that apoptosis was involved in the mechanism of action of these pectic extracts. In addition, to further confirm the involvement of apoptosis as a mechanism of action of these pectic extracts, we performed the DNA ladder experiment. And here is the result showed, in which we see in lane number 1 a marker of size of DNA, and then in number 2 the DNA of cells growing happily on cells growth media. In number 3, the cells were grown exposed to the pectic extracts, and we can see the fragments of DNA very well align with the effect of a positive control that we ran on lane 4. Lane 5 had no DNA and there is no signal on that lane. Another example of this strategy of finding bioactives on residues is what we did and published on 2020, looking on artichokes discards. When these delicious vegetables are put in a can, so we can enjoy it at any time, several parts are discarded, and this makes a lot of residues. So what we did again was treating them with different chemicals to get bioactive compounds. And what did we find on so with this artichokes extract? What we did was again viability assays, and we used normal fibroblasts, MCF7 cells, which represent breast cancer, and CACO2, which represent colon cancer. And at the beginning, the very lower concentration, there is no important differences in changes in viability for 3 cells used. However, starting at 500, we start observing a very important inhibition on viability for MCF7 cells. So there is selectivity with respect to normal human fibroblast. At the next concentration, CACO2 also showed a reduced viability, and fibroblast didn't show it. They had a viability over 100%. The same at the next, where we see further reduction in viability for MCF7 and also for CACO2 cells. So until this concentration, selectivity is maintained, and later on fibroblasts also started to show cytotoxicity. But there is a range of concentration in which there is selectivity for this extract that comes from a byproduct of the industry of artichokes. So in conclusion, I would like to say that despite these modern techniques available for drug discovery, natural products are still a valuable source of inspiration. So we can still be looking on traditional uses by different ethnias and find the inspiration to get new drugs that will save lives. And also the other approach that I showed, that is looking on byproducts of agro-industrial and other industries can be very important for finding new drugs. New drugs in different areas, including cancer, neurodegenerative diseases, autoimmune diseases, are still urgently needed. And just to finish, I'm going to show which is the road that these molecules have to go through. I've shown here preclinical assays, in vitro. Then, unfortunately, we need to perform the preclinical in vivo that involves experimental animals, and then go through the different phases of human studies. Phase 1, which look more for pharmacokinetics and safety, and then phase 2 and 3 that are progressively increasing the number of patients or volunteers in this case. Safety and efficacy in phase 2, we are still looking at the best dose or scheme of treatment, and then we do large multicentric studies that are called phase 3. And then at this point, if these molecules, let's say candidate 1, is successful, can apply to be approved or licensed for commercialization in the different countries. See, each country has its own regulatory agents that look at the data from the preclinical to the clinical, going through the different phases of the clinical essays. So to take a decision, a scientific decision on getting a new drug onto the market. Thank you very much for your attention.